Hello, and everyone who's watching this live or later on. We're going to be running through this paper 11 from 2014 in the AS Bio course. I have Maya here with me. So shall we just get into it quickly?
Yeah.
Okay, nice. Okay, so the first question. What best describes an electron microscope in comparison with a light microscope? So do you have any ideas on this at all?
Yeah, because like electron is like so small, tiny one, so it might be magnification might be higher.
Okay, that's cool. That's you've got it. So you don't have to know what I'm gonna tell you right now. So actually, do you know what resolution means in general?
It's like how clear it is.
Yeah, so it's actually what it means is that the ability to tell two particles apart, so two pixels, because it's, if you're using like a CCD camera image, then it's going to be 2 pixels. So obviously the smaller you go, the clearer the image is, the more detail you can. So if you have like, for example, if you imagine a resolution of like 1 centimeter, that's pretty big, right? And then you imagine like little dots inside there. If it's 1 centimeter and there's tiny little dots inside, if it's a 1 centimeter resolution, then that whole square or that whole radius is just going to look like one point, one giant point. And the problem with that is you're losing all of that information inside. So the limit for the light microscope is actually the wavelength of light. So there is an equation that you don't have to know. So if I just try and draw it, so that's an R. So the resolution can be given by this. So I'm just drawing it out so we can sort of go through why this is the case. Hopefully it will be helpful. So this symbol lambda means the wavelength of light, right? And these, what I'm drawing before and under, these are constants. So the one on the bottom, this is a property of the lens, and then this is a constant. So if you think about the fact that with visible light, so what you would be using in a light microscope, about as low as you can go is 200 nanometers. So the resolution that you can go is 200 nanometers. So the wavelength would be 500. That's the lowest you can go, because beyond that, it's not visible light. So if you use this equation with 500, and then the best lens that you can get, objective that you can get, would give you a value of 1.5, and like so you can get the 200 nanometer. But if you use an electron microscope, then obviously the wavelength of an electron can be way smaller, as you pointed out. So if you use the same equation, that can go down to about 2.5 over a different constant, because that depends on the electron microscope as well. And so that can go down to about 0.3 nanometers. So as you can see, that's also nanometers. You can get a much higher resolution on the electron microscope. So again, you don't have to know this. I just wanted to explain why, if that makes sense.
Yeah.
Cool. Okay. Should we go to the next?
Yeah.
Okay. Oh yeah, should probably also. Okay. So where would cisternae be found in a cell? Do you have any ideas? So do you know what any of these are at all?
Yeah, I know the second one and the third one. I'm not sure the first one.
Okay, so the endoplasmic reticulum, so they're all organelles. So what an organelle is a membrane-bound structure within the cell. And they all have different functions within the cell. So maybe without going into too much detail, for sure. So essentially, they're quite similar in the sense that they're all single. These two are bound by single membranes. This one's a double membrane. And then cisternae are sort of like flattened sacs of membranes. And the reason why they're separated is because they each inside each of compartment there is a different cellular process that's going on. So this is unfortunately one of the many questions that you either know or you don't. But yeah, so just for reference, the ER is connected to the nucleus. So a lot of nuclear products go into it. So translation happens in the ER, for example. That's one of the big ones. And it helps to create an environment in which proteins can be folded up into their correct shape. Does that make sense? Okay, so yeah, unfortunately, this one again is, you have to know, but I'm sure it will become clearer, once you go through it. Okay, next. Which adaptation would increase active transport of carbohydrates from a plant cell? Okay, so do you know what active transport is? No, that's fine. No, don't worry. So if you imagine a membrane, and on one side you have lots of particles, and on the other side you have less particles, then in which direction? So through this membrane, it's permeable, which means that through the membrane, the particles can cross. So some membranes are permeable, some aren't. So if this was the case, what do you think would happen? Do you think this would stay the same? So on either side of the membrane, there will be, on one side there will be more particles, on the other one less. Would it be eventually the same? Or do you think there would be particles only on one side? Which one do you think would most likely to happen over time?
Same.
Yeah, right. So if you imagine all of the particles are moving around, they all have kinetic energy, they're bouncing off, but they're going around. And obviously you can't say that this, for example, this particle will never go onto the other side. It will, but overall, because there's more particles here with more kinetic energy, overall, over time, they will indeed go this way. And then the result will be what's called an equilibrium across the membrane. So that's what would happen normally. But an active transport is normally something that requires energy. And so this is called a concentration gradient. And an active transport is something that, so it requires ATP or an energy because you're moving particles across a membrane against their concentration gradient. So it's not where they would normally, not the direction in which they would normally go. That's what it means. So in this case, should we just go through each one if you think this would help? So do you think, so it's across a membrane, remember? So first off, areas where the cell wall is thin, it's not really gonna help because it's not through the cell wall that's the problem, right? Okay, so increased permeability, what do you think would happen? That's fine, don't worry, it's fine. So if increased permeability would actually be bad for active transport because you're dissipating that gradient. So if you imagine this gradient, if you basically put more holes into it, then this process of equilibration will happen quickly, right? But you don't want that. You want the concentration gradient to remain, to basically put more particles on the concentrated side. So that wouldn't be good. And then a large surface area, the reason why this one would be good is because you have more space across which you can put your proteins that will then make the active transport for you. But it all requires energy. Okay. Sorry if I went into a little bit too much detail. We can leave it. I just thought, okay. Nice. So about this one, the size one, what do you think? Do you know approximately any of these, or why they are, what they are? Do you know any of these, like what they do or what they are?
Yeah.
Okay. Yeah. Do you wanna any of them?
Yeah, like I don't know if my pronunciation's
That's fine.
like nucleophile.
Yeah, that's perfect. You got it.
Makes ribosomes.
Yeah. It's within the yeah, it's yeah, so it's a space within the nucleus. But yes, exactly. You're right. So that's this one again is something that you need to know, unfortunately, you know. So this one can be 1 to 10 micrometers in size, depending on the cell. Okay, so do you know what a lysosome is? It's another organelle, membrane-bound little organelle.
Lysosome is. I'm not pretty sure.
But that's fine. So the way I like to think about it in simple terms is that's kind of like the digestive system of the cell. So what it has is it has a lot of digestive enzymes and a low pH. And so lysosome is just like a small ball, a small membrane-bound compartment. And when you fuse anything with it, it kind of eats up what's inside. Yeah. So, okay. And then the centrioles. So the centrioles, so do you know this what the cytoskeleton is? The cytoskeleton basically, so if you think about a cell that's an animal cell, for example, you know how when you cut yourself and then afterwards you get a swelling around the cut, you know, it becomes kind of red around it, it bulges up. So that's because you get your white blood cells that come to the area of infection. And the way that happens is that when they're in the bloodstream and they get a signal and they literally go through the vessel's cell between the vessel's cell wall. They squeeze through and they go to the site to fight off any infection. And they need to be flexible to do that, right? Because they're not really constrained by any walls. And the reason why they can sort of flatten out and go through is all due to the cytoskeleton. So the cytoskeleton, there are all of these tubes basically inside of the cell, and they can pull or push to change the shape of the cell. And also within the cell, they can help the movement. So if you think about all of these little compartments communicating with each other, the way they do that is through the cytoskeleton. So everything. So the centrioles are the origin of all of the tubes that keep the cell together, and then the ribosomes are the smallest one. So those are 20 to 30 nanometers. So do you know what those do?
Um, they're like small organelles.
There was sorry?
Small organelles.
Yes. So
like yeah.
That's fine. So ribosomes are useful in translation. So if you have your DNA that encodes a protein, then the way you get the protein is with the help of a ribosome. So the ribosome can't directly read your DNA. There's an intermediate, which is called an RNA. But what it does is it attaches onto the RNA, and then it essentially makes the protein. So it makes what's called a polypeptide. A polypeptide is like a unit name of a protein. So that's from each amino acid is the building blocks, and then that's what the ribosome does. It's literally just like reads it, gets the right amino acid, and then moves on to the next bit, reads it. So that's what it does. Is that okay? Is that overwhelming? Sorry if it's okay. But does that make sense?
Yeah.
Okay. Nice. So the next one. The diagram shows some similarities between chloroplast, mitochondria, and typical prokaryotes. Okay. So what do you think about this one?
Like I could know the number 3 is mitochondria.
Mm-hmm. And why do you think that's mitochondria?
Like, I don't know, like mitochondria is like, I'm not sure, I guess.
Okay, that's fine. So some organelles, chloroplast and mitochondria in particular, they have their own DNAs. So you know that most of our DNA is in the nucleus, right? But some have their own DNA and they can even replicate independently of the nucleus. So yeah, you're correct. So that one would be the mitochondria. And then the organelles which have their DNA, they have circular DNA, so that's fine, you know, the overlap. And then prokaryotes and chloroplasts also have circular DNA, and the reason why is because there's a theory that mitochondria and chloroplasts derive from prokaryotes. So they think that there was an event where mitochondria and chloroplasts were engulfed into a cell, and that's why they have a double membrane. So if you imagine them being membrane bound and they kind of get engulfed, then they are within two membranes. So yeah, that would be C. Okay. Should we move on?
Yeah.
Okay. This one, again, I'm sorry, but this is, again, a classic one that you need to know, unfortunately. A student carried out 4 tests for biological molecules on a solution. The results are shown in the table. Okay. So do you know anything about these? It's fine if not, I'm just asking. Okay, no worries. So iodine solution, this one is a test for starch. So if you put in your sample, all you want to know is what's in it. So if you put in the sample, iodine is brown, and if there is starch, then it turns sort of blue-black. The solution turns blue-black. So in this case, it didn't. So we know that there's no starch in it, right? Okay, the next one. This one is a test for proteins, and it turns purple when there is proteins. So we know that there is proteins, so that's good. No starch. Protein. And then Benedict's tests for reducing sugars. So that means, so starch is a polymer of sugars. So it's like one long molecule of repeating units of sugars. But every, it's called a monosaccharide. So every, you know, the one building block, they're reducing. So that means, should you know what that means? I need it. Okay, perfect. Yeah, exactly. So that's literally what it tests for. Exactly. And then some disaccharides are also can be reducing. So in that case, it turns orange. So that's good. So we know that there is reducing sugars. And then the emulsion is a test for lipids, and it turns milky when there's lipids. So we know that there's no lipids. Okay, so based on this, what do you think, which one do you think it could be correct? So we already know that this one can't be correct because there's starch, right?
Mm.
Okay, so collagen is a protein, right? This one can't be correct either. And sucrose is not a reducing sugar. So therefore, this one is the only one that, okay. Oh, speak of the devil, here's the, the polymer. There we go. Okay. So this one, if you don't know the reactions, then this one is a difficult one to know. But I think, you know, don't be alarmed if you don't know it, because these are just like, you know, very protocol reactions that you will meet again and again and again. So I'm sure that it won't be a problem after a while to remember them. Yeah, so in this case, you want to test if there's any reducing sugars in the molecule, right? So yeah, you just look at from a protocol. you hydrolyze the molecule and then you test for Benedict's. Yeah. So that would be this one. Should we do the next?
Yeah.
Okay. Which molecules show the structure of alpha glucose and beta glucose? Okay. So have you heard about this before?
Yeah.
Okay. So can you tell me which one and why?
I don't know, but like why it is, but because just I remember it. I remember it. Yeah.
But okay, so what should I look at in the molecule that determines whether it's alpha or beta?
Oh, I remember like there.
Yeah, that's perfect. Exactly. So this part. Yeah. Is that what you were going to say?
Yeah.
Okay, good. Okay. Yeah. So the way you can tell is that if you imagine this, so like this hexagon, if you look at my hand kind of like just horizontally, and then you have either one of these groups, so this one or this one, sticking out from up or down, right? And you always look at so you start counting, there's 6 carbons, you start counting the carbons next to the oxygen, so like that, 1, 2, 3, 4, 5, 6. And the way you look at it, by convention, is that you look if this hydroxy group and this CH2OH are on the same side or not. If they're not on the same side, then it's alpha. If they're on the same side, like here, it's beta. That's it. But yeah, you got it. Cool. Okay. In unsaturated lipid molecules, where are double bonds located? Okay. So what do you think? So I see you have a diagram of
yeah.
That's pretty good. Okay. So I'll just draw a simplified diagram like you have there. I'm not gonna draw out the molecular structure, so I don't want to confuse you. Okay, so I see you've got the diagram. I'll draw the same.
Yeah.
Okay, so that's the glycerol, right?
Yeah.
And then you've got your fatty acids here. Ooh. There we go. Okay. So the answer to this is that you want so an unsaturation means double bond, okay? And you want basically, so have you ever heard like when you buy olive oil or something, or some good healthy oil, it says unsaturated fatty acids inside, right? And the reason why so you can see that most of those products which have polyunsaturated fatty acids are liquid at room temperature, right? So butter also has a lot of lipid molecules, but it's solid normally. And the reason why that is if you have a double bond inside the fatty acid, then you would have a kink in the tail. So it would look something like that. So it would be like straight on, and then where you have a double bond, it would come out like that. And then the reason why this means that then the lipid will be liquid is because the molecules can't pack together as tightly as they can when it's saturated. Does that make sense?
Yeah.
Okay. So there will be within fatty acids. Okay. And then glycerol always stays the same. It's that's the backbone. It doesn't really change. It can't. Okay. I think this one's a simple one, for sure. So this one, you just have to basically add up the charges and see if you get any leftover charges. So what about, glycine? What do you think? Does that have an overall charge? You're not sure? Or?
No, sure.
Okay, so you have just a negative charge there and a positive there. So those ones sort of cancel out. and that would have an overall 0 charge, right?
Yeah.
Okay, minus, and then this one, you have a negative there, positive there, positive there. So this one has an overall positive charge. What about this one?
It's like 0.
Yeah, perfect. And this one? Okay, so it has a negative, a negative one means -1.
Yeah.
Otherwise it would have a number. So -1 plus 1 is 0, and then you have one more negative there, so overall negative. So it's these two then. Does that make sense?
Yeah.
Nice, okay. Okay, which level of organization of the hemoglobin molecule is coded by a DNA molecule? Okay. So do you know what all of these mean? The primary, secondary, tertiary, quaternary structure?
Yeah, I can imagine, but...
Okay. Do you want to try to talk about it? Like, you don't have to say all of it, just whatever you know or whatever you're thinking about.
Yeah.
Okay.
Sorry.
That's okay. That's no worries. So hemoglobin, do you know what type of molecule is it? It's a protein.
Yeah.
Yeah, okay. So, or a polypeptide is a smaller unit. So do you know, so the building blocks of a protein, do you know what those are called? Amino acids. So primary structure would be sort of just what kind of amino acids they're made up of. That's all. So if you go, for example, like these ones are examples of amino acids. So primary structure would be, I know that this code has a glycine, then an alanine, then a lysine, and then an aspartate. That's it. There's no other information. A secondary structure is, so all amino acids, do you know sort of what an amino acid looks like, what the common structure is? Because I think if I draw it, then it might help to understand it. Do you think?
Yeah.
Okay, so let's do NH2 like that. Okay, sorry. Here we go. Okay, so this is a basic structure of an amino acid. The R means that this is what varies. So the different names of the amino acids come from the different groups. So this always stays constant. So for example, if we go back to the previous question, like that, you can see this is the constant in aspartate, and then this is the R group. So can you see that this is the same as this? Does that make sense? No, you can't. Okay. Okay, maybe I'll draw it up here, and then we can we can try again. Okay, hold on. Let's do it like that. Okay, I think that's better. Okay. I'll draw it like this because that's more okay. Okay. So if you just look at, this is a shorthand, just like COO. The shorthand means that it's not fully written out what the molecular structure is. That's connected to a C, that's fine, which has an H coming off of it there. And then you have the NH3. And then this group is the one that varies, which is the R. Does that make sense? About more like this? And it's exactly the same here. So if you look at it, there's your center, there's your carbon. Can you see that that's exactly the same as this? Yeah. And then exactly the same here. This one is all the same. And then this one is again the same. And the only thing that varies is this, and that's what we call the R group. And that's how you get the primary structure is these. But then the secondary structure is essentially these two can have chemical interactions with each other. So sometimes they can form like a helix. And then the tertiary is the interactions between the R groups within a protein. And then finally, the quaternary is interactions between polypeptides. So polypeptide means that it's only one molecule. It can have a really different structure, but a quaternary is separate polypeptides interacting with each other. So for example, hemoglobin is made up of 4 polypeptides that interact with each other. So what is actually coded in the DNA molecule has to be the primary structure, because everything else is determined afterwards. So it's a chemical, so that the DNA is not going to tell you, which R groups will interact with what, right? Because that's not contained in the information. All that your DNA is going to tell you is which amino acid you're going to have, right? Okay. Okay. So these statements are about enzymes. They're globular proteins. They're formed in the smooth endoplasmic reticulum. They're only found attached to plasma membranes, and they can be inhibited by competitive inhibitors. So this one is, again, unfortunately, something that you need to know that you can't really, but yeah, so do you know at all any of these, or which one might be true?
Yeah, I knew the 4 is the answer.
Yeah, perfect. Yeah, that's definitely one. And then this is not true. Like, you can have enzymes anywhere. You can have enzymes, there's enzymes in your saliva everywhere. So they're not limited to the membrane at all. They're not formed in the smooth endoplasmic reticulum. And yes, they are globular, which means, do you know what a globular means? So globular means that they are soluble in water. That's it. So they have, so some amino acids can be hydrophilic or hydrophobic. Hydrophilic means that they're soluble in water. They like to surround themselves with water. And hydrophobic is exactly the opposite. So if you think of like oil is really hydrophobic, if you pour it into water, it just forms like a blob. So that's exactly what it is. So when you have hydrophobic amino acids, they group together. So enzymes are globular proteins. Cool. Okay. Yeah. Okay?
Yeah.
Okay. An unusual enzyme has been found in a tropical grass. It catalyzes the hydrolysis of the fungal polysaccharide chitin into amino sugars. It also inhibits the activity of an enzyme in locust guts, which catalyzes the digestion of amylose. Okay. So what describes the actions of the enzyme? That's fine. Don't worry. It's fine. It's okay. Look, all the reason why I'm asking is if you have any ideas. Like, it's completely okay not to know the answer. I just want to have a sort of a discussion. If you have any ideas about it, don't feel bad if you don't know the answers. That's exactly why we're here. Don't worry. Seriously. Okay. So this one is actually a kind of a strange question because, so a polysaccharide, again, is, it's similar to that molecule that we saw is like a repeating unit, a really long chain of molecules. And so is amylose. That's also, amylose is a component of starch. So again, that's exactly what we saw up in that question with the polymer. So it looks like that. So it can be broken down into individual building blocks. And the way you do that is, so a glycosidic bond, that's a bond between, so that's a glycosidic bond. Glycosidic bond is a bond between the monomers of a long chain like this. So a hydrolysis means that the way you break this up is essentially, the reason why it's called hydrolysis is because you're adding water. So you're adding a water molecule to break this bond up into its individuals. And so the reason why this is strange is because this enzyme, it both catalyzes the hydrolysis of glycosidic bonds because it breaks down this polysaccharide into single sugars, but at the same time, it inhibits the enzyme that would break down amylose. So it both catalyzes the hydrolysis of glycosidic bonds and inhibits, but they're different glycosidic bonds, so therefore it's not a problem. Does that make sense? So it, at the same time, it hydrolyzes but also inhibits, but they're different reactions. It's just a similar bond. Okay. The fluidity of the cell surface membrane can be changed by a number of factors. As the fluidity of cell surface membranes decreases, which process would be least changed? Okay. What do you think of this?
Like, I didn't get like the meaning of this question, so.
Okay, that's fine. So a membrane is not really a solid, right? Because it's really malleable, it can be changed. The proteins inside the membrane, they're not static, they can move around. So one of the ways to make it sort of more liquid is, we've already seen that, you can have unsaturated fatty acids, for example, that will make it more liquid. And in this one, if you have a tighter membrane, then you're not gonna have, so the diffusion, do you know what diffusion is? It's exactly what we've already seen. So if you have a membrane, it's the movement of particles across a membrane, right? So if you have a tighter membrane, it's not gonna be as easy for the particles to go through it, is it? So this one would definitely be changed. And osmosis is a kind of diffusion, it's a diffuser. So that's the same. Endocytosis is the engulfing of something from the outside of the cell. So it can be like a pathogen, like a virus. It can be a number of things. So if you imagine like a cell like this, and you have like, I don't know, whatever, it's like a virus, let's say, then what the cell can do is it can indent like this, and then eventually it can, oh, okay. You can just have like a little particle with the, for example, with, okay. So if this happens, if you have a rigid membrane, this is not gonna help, is it? Because it has to sort of move in towards itself. So if you have a solid one, it's not gonna, it's not gonna be as bendy. But then again, active transport, that's in a way independent, not completely independent, but it's more independent of of what the membrane is like, because it depends on the protein. So the only job it does is it moves particles across the membrane, but it's through a protein, so it's independent of what the membrane looks like, right? So that would be active transport. Okay. The next one, do you have any ideas which molecules contributes to cell recognition in cell surface membranes?
I looked up, and like there's a word like net movement. I don't know if it's, but I didn't understand what is net movement.
Net movement but this one is about the recognition, right?
Yeah.
So the net movement is, we've already seen that happen. So do you remember when we talked about how if you have a concentration gradient, over time it equilibrates across the membrane? So net movement means that even though some particles can move across the membrane from either side, over time, a net movement is down the concentration gradient. That's what it means. Okay.
Yeah.
So this one is gonna be C, because cholesterol is a part of the membrane which helps with its structure, its fluidity. Phospholipid is the building block of the membrane. And then, yeah, so this one is really the only one that makes sense, because it, by process of elimination, you can't have cholesterol or phospholipid. And then, yeah, so glycolipid and glycoproteins, they are modified parts of the membrane. So they're modified lipids and proteins which are in the membrane, basically. That's it. And then there's so many different kinds of protein that do a number of different roles in the cell. Okay?
Yeah.
Okay. Do you know what facilitated diffusion is?
Yeah.
Okay. So facilitated diffusion is just diffusion, like we mentioned before, but it happens through a protein. That's what it means. That's it. So it's different from active transport because it's not a movement of particles up their concentration gradient. It's still the same. It's still down the concentration gradient. The only reason why it's different is because it goes through protein. So a scenario in which this would be useful is if a molecule wouldn't otherwise go through the membrane, you can put in proteins that help it go through the membrane. So for example, like a membrane is quite hydrophobic. So you can literally put in a protein that has a channel through it, through which the water can go through. And then it kind of, acts as it helps sort of connect the inside and the outside. And then, it lets some particles flow through it. But it's not active in the sense that it doesn't use energy to move the particles up their gradient. Is that okay?
Yeah.
Okay, cool. So what do you think about this? Yeah, okay. Yeah, so that would be exactly, they just use membrane proteins for both. Okay. Okay, unfortunately, this one again, it's one of those. I know it's difficult because I like to do biology in the sense that it makes sense for you to understand, but this one again, it's one of those that you look up and you know what happens. Yeah, so you can think about having many mitochondria in a cell. That means that it's going to have a lot of energy, right? So you probably want active transport there because you probably have more ions in a cell, right? If you want to take it up. So if you want active transport, then you need a lot of energy, which means that it will have a lot of mitochondria in that area, if that makes sense. And therefore, you also want the protein carriers there to take it up for you. Vacuole kind of irrelevant in this case because that's an organelle inside the cell. So it doesn't really have any effect on the take-up of ion uptake. Okay. Do you know this one? Do you have any ideas?
Like I don't get it about like plasma cells.
Mm-hmm.
Yeah, I read like some like definition, but I couldn't understand.
Okay. I think what the question is getting at here, this one. So I think it's talking about blood plasma, I think. So they're essentially, if it's not specifically said that it's a gametic cell, then you can either have a gametic cell or an autosomal cell, which is every other cell in your body, other cell in your body, and they all reproduce with mitosis, unless it's a gametic cell. So a cloning would be via mitosis, and then as we just said, the gametic one isn't. That's meiosis. That's a very specific one. And then also, you could replace dead cells. You could replace by clones. So just use mitosis. Okay. Okay, should we go through this one?
Yeah.
Okay, so do you know what this means? The 2N, or what the N stands for at all?
Yeah, it's nuclear.
Yeah, so N stands for the number of different chromosomes in a cell, right? So you know when a gamete is produced, the reason why meiosis happens is it halves the amount of chromosomes that it has. So you're still going to have every kind of chromosome, but you're only going to have one copy of each. Does that make sense?
Yeah.
Cool. Okay. So if you imagine this one will have, so the way to get a hybrid is you need two gametes to fuse, right? So these two gametes will have half the amount of chromosomes. So 7, right? And then this one will be 14, just like that. So mutation doubles the chromosome number. So that just means the overall chromosome number. So we know that 2N is every individual that isn't a gamete. So that just means you, me, you know, so that would just be, if it doubles the chromosome number, it would still be 2N, but now it would be double. Okay? Then, if you have a think, this plant would have, what would its gamete look like? How many chromosomes would the gamete have? Yeah, exactly. No, you've got it. And then this one is still 7, right? So again, then you have the offspring, kind of weirdly 21. Ooh, like that, right? And then this mutation, again, just like before, doubles the chromosome number. So you get 42. Yeah?
Yeah.
Okay. So diagram shows a human cell at the end of prophase of mitosis and the daughter cell just after telophase. How many DNA molecules are there in the nucleus of cell 1 and cell 2? So what do you think? Let's just go through. So how many are there here?
In cell 1?
In cell 1, yeah. So it's just a normal human cell.
So 40.
Yeah, exactly. And these two are daughter cells, and they go through mitosis. So we know that mitosis yields clones that are identical. So they have the same number of chromosomes. So these are also each 46. So yeah, if you add those two, oh my God, sorry, because it's going through mitosis still. So eventually, the daughter cells will have 46. Sorry, I messed up. That's my bad. Anyway. So at the end of prophase cell 1 and then the daughter cell just after telophase. Okay. Sorry. Yeah. Anyway. So yes. Okay. Let's go through it again. Okay. Anyway, sorry. So so mitosis. Yeah. Okay. I know why I messed up. Don't worry. Okay. So the way you get mitosis is the way you get from 1 cell to 2 cells is you need to double the amount of your DNA first, right? Because if first it divides, then it will have half the amount of DNA, and you don't want that. You want it to be a clone of your first cell, right? So, sorry, I misread the question actually. So, at the end of prophase of mitosis, this means that at prophase of mitosis, you have already doubled your amount of DNA, okay?
Yeah.
So cell 1 will have double the amount of normal DNA with 92, yeah. And then this one is still exactly what we talked about. So these ones are at the end, once they've split down, and these remain 46. Exactly. So sorry about that. I misread the question. Okay. But that makes sense.
Yeah.
Okay. Which diagram represents the correct base pair of DNA? So can you maybe walk me through, so I see you have this one also highlighted, so that's correct. So can you tell me why these ones are not correct at all? So what are some things, do you know anything about DNA? Like what are some fundamental things that have to happen for you to get a good structure of DNA?
I don't get like these like, is it like shape is different? Is it like some meaning of like these shapes?
Okay. Oh, you mean like this one is sort of like a dome?
Yeah.
Okay. Yeah, so this one just means that these always pair up with each other. So like you can't have a C and a T because they don't fit together. They don't obviously look like that, but that's what it means. Okay. Okay. So let's just go through each one. So why do you think this one is incorrect? Do you have anything at all that kind of comes out to you? It's fine if not, again, I'm just asking if you know. Okay, so what if I tell you that A and T always link up with 2 hydrogen bonds, and then G and C always link up with 3 hydrogen bonds? So that's incorrect, because they can't form 3 hydrogen bonds, so this structure can't happen. Okay, this one, they do have 3, so that's fine. So why do you think this is incorrect?
This.
Yeah, exactly. So what this calls, so the way, if you think about it, a DNA, I should probably write on the one that's correct. So a DNA strand, they're called anti-parallel. So if you, for example, if I was to draw another base pair underneath, like that, let's do A. Very ugly one, but that's fine. Like that, okay? So this is the only way in which they can link up to one another. So this just doesn't exist. So a nucleotide always looks like this, always. So you can't have a mirror image. So if you imagine this side looks exactly like that, but they're running opposite directions. So if you were to flip it around, you would get the same thing, like rotational wise, right? So this one just doesn't work because you can't link up the base pairs. And then this one is exactly the same. Why it doesn't work? Also, it has 3 instead of 2. And then that one is the correct way. Okay? Okay. Part of the amino acid sequences in normal and sickle cell hemoglobin are shown. Okay. So here are possible mRNA codons. Okay, so do you know anything about this at all?
No.
That's fine, don't worry. Okay, so do you remember how I said to get a protein from a DNA sequence, you need to go through RNA? So all it does is you have your DNA like that. I don't know, I'm just gonna write whatever, GCC. And then what it does is opens up, so here would be the pairs. Sorry if it's ugly. And then what would happen is if you wanted a protein, then this would open up like that. And then an RNA molecule would be synthesized. So it would just be this small, it wouldn't be as long as, because this one goes on, obviously. So, yeah. One additional complication is that in DNA, you get AT pairs, but in RNA, you get AU pairs. Sorry about that. That's a bit annoying to remember. But essentially, if you see U, you can think about that. That's the same as T, but in an RNA. Okay, so you get TA, right? G, that's still pairs with C. Then you get GG. And then this gets transported. And then this one is then, as previously we talked about it gets recognized by the ribosome. And then the ribosome, what it does is it grabs these tRNA molecules like that. And for example, let's take the same strand, right? So U, A, C, G, G, right? From up there. And what it does is, here's the ribosome over it, and it puts the corresponding tRNA. So if you have UAC, it would grab a tRNA with AUG, right? And the tRNA brings an amino acid with it. So this is exactly just how you build a protein, is you have these there's three, so they're called triplets, and they each code for one specific amino acid. And the way it does is exactly just with the help of the ribosome, it brings the tRNA there, and which with the amino acid, and then it moves on to the next one, brings the amino acid, and then you sort of build it up, building block by building block. Is that okay? Okay, cool. So here we've got our amino acids. So these are the codons. So if you imagine what would be the RNA sequence to this, so just literally pair it up. So a C pairs up with G, yeah, okay. So A normally pairs up with a T, but if it's RNA, U, yeah, exactly. Yeah, okay, so this one. Yeah. 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 Exactly. So now we can have a look if these are up here. You've got a valine, GUA, that's good. B is GAA. ACU is threonine, and ACG is nowhere to be found. So therefore, this one is not involved. Okay, yeah. Great. Two sets of bacteria were grown using different types of nitrogen-containing growth media. One set was grown in a medium containing the heavy isotope of nitrogen 15N until all the DNA was labeled. The other set were grown in a medium containing the light isotope of nitrogen 14N until all the DNA was labeled. The DNA from each set of bacteria was extracted and centrifuged. The diagram shows the position in the centrifuge tubes of this DNA. Okay, so 14N is lighter than 15N. That's pretty straightforward, I think, right? And then the bacteria with 15N labeled DNA were transferred to a medium containing 14N and allowed to reproduce once. The DNA of the new generation of bacteria was extracted and centrifuged. Which tube shows the position of DNA? Okay, so what do you think? So we know this length is the 15N one, right? Or 14N, sorry. 14? 14N, yeah. 14N. And then this one is the 15N. So which one do you think would be
What is the centrifuge means?
So if you put something, so centrifuge is, it kind of, it's like, if you look into it, it's like a disc, and you put your tubes inside it, and it spins around in a really, really high rotations per minute. And what it does is it can separate out, if you put it into the correct solution, it can separate out things based on their density. So this is exactly what happens. So if you have something more dense, it goes towards the bottom, and then if you have something lighter, it goes towards the top. So it just means that anything that's heavier will go down, and anything that's lighter will go up. Okay, let's go through it anyway. So this is the correct answer. So this is sort of halfway between these two. And why do you think that might be the case? Because, okay, so the question just said that if you start, when we started out, right, all of the DNA inside the bacteria were 15N, right? All of it. If you go back up, that's what it says. One set was grown in a medium until all the DNA was labeled, and then they were transferred to 14N, and they were only replicated once, right? So this is what you start out with, that's fine. But then, so you know the way DNA is replicated is called semi-conservative. So the way you know, you want 2 DNA strands from 1, and the way you do that is you separate out the strands like so, and they become templates for your new strand. So all of the bases that I've drawn in red come from the medium that the bacteria are grown in, right? So that means that all of these are going to be 14N, right? But the original is still 15N. So we are explicitly told that they are allowed to reproduce once. So this is all of their DNA looks like this at this moment. So this is why when you separate their DNA out, they will have a weight between the two extremes, between 15N and 14N. But for example, if you were to let them grow another time in the same media, then you would see what would happen. This is not part of the question, but I think it helps to understand, like, hopefully. So now you have these 4 as your templates, right? Oops, sorry. Like so. But your new strands also come from 14N, right? So if they were let to replicate one more time, then you would have some which are here, which correspond to these 2, and then some which are already back up at 14N, right? And if you were to do this, many more times in the medium, then eventually you would lose the 15N signal. Does that make sense? Okay. Which processes are involved in transpiration? This one is, again, I won't spend a lot of time on it because so transpiration is the process of water vapor leaving the leaves. So there you go. We've already got one, diffusion of water from the stomata. Stomata are the cells that, open and close. Okay, that's fine, and then the mass flow of water through the xylem. If you imagine a molecule leaves from the top, all of the water molecules are sort of in contact with each other. So if one leaves, it sort of pulls the next one up, and that means that the entire water column will move up a tiny, microscopic bit up. And then, the evaporation of water they evaporate from the air spaces between, not the surface. Yeah, that's fine. Okay, let's move on. Okay. Once again, don't worry about this too much. This one is, again, a classic one of those things that if you've seen a cross-section of a stem, then you will know which one is correct, but don't worry about it too much because I think it just takes a while to, be sure to be able to identify each component. So that sieve tube is like a porous structure. So that would be X. And then that one is the xylem. You can see that it has a really thick wall. So it's like a thickened wall because that gives structure to the vessel inside of the plant. And yeah, and then that's it. So okay. The graph shows the results of using a photometer to measure the effect of wind speed on the rate of water loss in a plant. Okay. Which statement explains the results obtained from this investigation? Okay, what do you think about this?
What is photometer? Like,
yeah, so I think it just measures the rate of movement of, so like the loss of water. That's all it does. But even if you didn't know that, I'm sure you can describe the graph. So you can see that even without reading anything, you can see that the rate of water loss increases with the wind speed, right? But then eventually it closes off. So the plant is obviously, they respond to environment just like we do. And one of their ways to prevent water loss is the closure of, the pores, the stomata that we talked about immediately. So if they sense in the environment that they're losing too much water, then one of the ways in which they can counteract this is close it, and then it will drop back down, right? So yeah. So that would be C. Okay? Land flooded by the sea is not suitable for growing plants after the salty flood water has drained away. Which values of water potential in the xylem and soil water help to explain why the land flooded with salty water is unsuitable for growth of plants? Okay. So for this one, I think you should probably know that the flow is from high water potential to low water potential, right? So if you have a much lower water potential in the soil, you ideally would like your water to flow into the plant, right? Not out of it. So if you have flooded the soil with salty water, you are left with the salt in it, which will lower the water potential. So you get the exact opposite effect. So you're sort of drawing out the water from the plant instead. So yeah, in all the other cases, you would get the flow into the plant. But you can see that this one is more negative than the xylem's water potential. So there you get, yeah. Okay. Okay. We can go through the ones. How about we go through the ones that are recall? Just skip through those and then go through the ones that are concept. How's that? Okay. This one, definitely one that you need to recall, unfortunately. Yeah, and we can talk about this one a little bit, just briefly. So an artery carries, what do you think, high or low blood pressure? Blood with high or low pressure?
High.
Yeah, exactly. So an artery needs a thick wall, thick and muscular wall to be able to maintain the high blood pressure. So you've already answered the question perfectly. Okay. Yeah, so muscle and elasticity. Okay. Okay, this one more time. Another recall one. But yeah, so this one, just quickly to go through. So hemoglobin Hb is the component which can carry your oxygen. So in the alveoli, that's the site of gas exchange, where you're taking up your oxygen. So you get oxygenated hemoglobin. Okay. This one, what do you think? This one, I think we can go through pretty simply, as we talked about what requires to be, inside an artery. So what do you think of this?
First, like we could know like W is artery.
Yeah, thick wall, once again.
Thick, so.
Quite, the circular because of the high blood pressure. So yeah, exactly. And then that's kind of, it looks more limp and more thin. So that's the vein. And then, yeah, okay. So we know X doesn't contain oxyhemoglobin because oxyhemoglobin is the oxygenated blood, which is in the artery, right? Okay. Nice. This one more time, something you need to recall. Sorry about that. But yeah, basically goblet cells, they don't produce enzymes that destroy bacteria. They're between the cilia, and they are swollen with the mucus that helps sort of get rid of the bacteria. So they're indirectly involved in destroying bacteria, but they're not producing the enzymes, if that makes sense. Okay. Which could lead to chronic bronchitis. So if you think about bronchitis, sort of like a really bad cold, what do you think of this, if this one could be? It's fine if you don't know as well. This one is another recall one, I think. Yeah, so if you produce an excess amount of mucus, then, so the cilia are the cells, they're sort of like hair-like cells, and they help move the mucus, which traps the bacteria out of your lungs. So when you're like coughing up, then you're coughing up mucus that has, the bacteria in it or the infection in it, and it helps clear up. So if you are you have too much mucus, then it can almost sort of suffocate the cilia cells. So therefore you can get sort of like chronic, exactly. So like overproduction of mucus, but it's like a vicious cycle because you're not getting that cleaning effect. Okay. Almost there now. Yeah. Okay, this one again, if you don't know what an alveolus looks like, it's difficult to count. So I think I would leave this if because it's easier if you have a diagram to sort of count through. So let's go and discuss stuff that we can, also about TB. It's another infectious one. So should we just skip through the infectious questions? Because they are recall. Okay, let's go to the phagocytosis one then. So if you just go through in your head, sort of, do you remember that diagram I showed you where there's like the little virus on the outside, and then, so what do you think this one could be? So endo means inside, and exo means to release to the outside. So what do you think this could be?
So maybe like endocytosis.
Yeah, that's definitely gonna be in first, for sure. Yeah, good. So actually that's already easy. But okay, so let's go through it anyway. So the vacuole is the membrane bound thing that sort of forms afterwards. So if you imagine your membrane sort of coming in, and then once it's pinched off of the membrane, then you have, that's the vacuole. And then enzymic digestion. So for example, with the lysozyme, you could fuse with the lysozyme, you could digest it up. Yeah, and then Yeah, digestive, and then exocytosis, you release the rest of the products if you don't need it. Okay. Luckily, hopefully that makes sense. Okay, so which type of immunity occurs as a result of the ingestion of antibodies by an infant through its mother's milk? What do you think of this? So I think you can already see definitely not artificial like a jab would be, a flu jab or something like that. And then it's passive because there is no current infection involved, right? So it's just literally passing all the antibodies that are, okay. Okay, so this one, again, is something that you, but let's go through it anyway. So I know you probably haven't heard of this, but don't worry about it. So habitat, so okay, so let's read it out. An insect lives in and feeds on the tissue of an oak tree leaves and is eaten by birds. Okay. So a habitat is the environment in which something, an animal or a plant lives. A niche is like specific environmental conditions in which it can live. So so by the fact that it lives in and feeds on the tissue of the oak, it's a very specialized role within, you know, the ecosystem. And then the trophic level means sort of where it is in the food chain. So then, you know, we basically get information from all three of these. So you can take all three. And then finally, okay, the farmer grows a different crop in the field each year for 3 years. In the 4th year, the farmer plants a leguminous crop and then plows this into the soil. The next year, the rotation starts again. Which microorganisms will increase by the time the rotation starts again? Okay, almost done. So do you know anything about this?
No.
Okay, so I'll just quickly say that there's lots of different kinds of bacteria that can help with the soil. Decomposing one is, I think, an obvious one because, you know, that helps break down any sort of organic matter, be it, you know, like our flesh or, you know, plants or animals remains. And plants require the help of bacteria to fix nitrogen. So we use nitrogen as well, but they need sort of like a symbiotic relationship with bacteria to fix the nitrogen for them. So yeah, so you wouldn't get any denitrifying bacteria. That's pretty much it. Yeah. Okay.